We are advancing a new class of broad-spectrum cancer therapeutics that are specifically designed to treat some of the most aggressive forms of cancer. My name is Zeno and I'm the CEO of Hemispherian. We are an ambitious young team of scientists and engineers aiming to improve cancer treatments with innovative new therapeutics. It is well established that depletion of the DNA base 5-hydroxymethylcytosine in genomic DNA is a hallmark of cancer. For simplicity, in this presentation I'll refer to 5-hydroxymethylcytosine as 5-HMC. In effect, the less 5-HMC present in genomic DNA, the more aberrations in the epigenetic regulation of cell division, often leading to cancer formation. A critical finding by my colleagues Adam and Teresa was to establish a link between cell doubling time and the genomic content 5-HMC. As you can see on the left, the quantity of 5-HMC is directly proportional to doubling time. Cell types with a long doubling time, such as brain cells, have a relatively high amount of 5-HMC, whereas tissues with rapid doubling times, such as spleen and testes, have a relatively low amount of 5-HMC. On the right side, we see that normal cells have a relatively high amount of 5-HMC. As these levels decrease, the cell becomes precancerous, and then at even lower levels, cancerous. Adam and Teresa wondered what would happen if they increased the levels of 5-HMC in cancer cells, in effect pushing the cell from the cancerous region into the more normal region. Based on their research, they hypothesized that increasing the 5-HMC levels would result in a cell cycle arrest. To test this hypothesis, we overexpressed the enzyme responsible for the deposition of 5-HMC on DNA, that is TET2. As expected, this resulted in an increase in genomic 5-HMC levels and a cell cycle delay. Another interesting observation was that there was a threshold of TET2 expression that any given cancer cell could tolerate. TET2 expression and 5-HMC levels above that threshold resulted in cell death. These discoveries led to an interesting revelation. Cancer cells are depleted of 5-HMC. Therefore, they have a lot of substrate for TET2. In contrast, normal human cells are fully saturated for 5-HMC. All sites that could be acted on by TET2 are effectively blocked. Based on the ability to genetically target this mechanism, we reasoned that if we could increase TET2 activity with a small molecule, this small molecule would selectively kill cancer cells and would have no effect on normal cells. We then began to look for small molecules that enhance TET2 activity. Surprisingly, other groups had already discovered a molecule that enhances the catalytic activity of TET2. This molecule is vitamin C, which is a cofactor of the TET2 enzyme. Treatment of cells with vitamin C results in a significant increase in genomic 5-HMC levels in cancer cells. The effect of vitamin C was also evaluated in xenograft models. Tumors in mice that are treated with vitamin C showed a significant reduction in tumor size. A different group therefore initiated clinical trials treating glioblastoma multiforme patients with high doses of vitamin C. In these clinical trials, treatment with high-dose vitamin C resulted in an increase in mean progression-free survival of more than 6 months and an increase in overall survival by almost 7 months. We can therefore conclude that targeting of TET2 with molecules that increase the activity of TET2 significantly improves the outcomes. This validates TET2 as an ideal drug target for the treatment of cancers. We designed a biochemical assay to screen for molecules that can enhance the activity of TET2. This assay revealed 6 molecules that enhance the activity of TET2. Of these molecules, 2 were especially good at increasing TET2 activity. These molecules were then named GLIX1 and GLIX5. We evaluated the ability of GLIX1 and GLIX5 to specifically kill cancer cells in vitro. GLIX5 is cytotoxic to a broad range of cancer cells, whereas GLIX1 is cytotoxic to a much more narrow range, specifically glioblastoma multiforme, as seen in the middle panel. A good drug would be highly cytotoxic to cancers while being poorly cytotoxic to normal cells. GLIX5 has some cytotoxicity towards normal skin and kidney cells, whereas GLIX1 had no detectable cytotoxicity in any normal cells that we tested. We have verified the efficacy and tolerability of GLIX1 and GLIX5 in mouse xenograft models. Tumors in the group that received no treatment grew large and had a negative impact on the mouse. The group treated with GLIX5 showed tumor regression in 8 out of 8 cases. The group treated with GLIX1 also showed regression in 8 out of 8 samples. However, interestingly, 4 out of the 8 were regressed to the point that no tumor was detected. We also verified that the drugs were killing the xenografted tumors by targeting and enhancing the activity of TET2. We found that genomic 5-HMC levels were relatively low in tumors from the control group. In contrast, the tumors treated with GLIX1 had a significant increase in their genomic 5-HMC levels. This indicates that the correct mechanism has been targeted. We presented these results to the Chief of Neurosurgery at the National Institutes of Health in the US. He evaluated our compounds using neurospheres created from human glioblastoma patients. According to him, this model system is the most accurate representation of treating humans. In all cases tested, GLIX1 and GLIX5 were cytotoxic to the patient-derived neurospheres. Another very important property of a good cancer drug is that it is well tolerated. Treatment with GLIX1 or GLIX5 had no effect on mice's ability to gain weight, indicating no negative side effects. The mice even continued to gain weight when given extraordinarily high doses of up to 2,000 milligrams per kilogram. Furthermore, we noted that treatment with GLIX1 or GLIX5 resulted in no adverse effects on mouse behavior or vital signs. Given our PK values, we note that the anticipated human dose is 100 to 200 milligram per 70 kilogram individual, making human therapeutic doses very far below the maximum tolerated dose. As mentioned earlier, we have multiple compounds in our pipeline for treating various types of cancer. At the moment, we are advancing GLIX1 into preclinical trials. We anticipate that these will be completed within 18 months. GLIX5 is currently being optimized. We are assessing synthesis routes and potential variations of the compound itself. We have 4 other compounds in the GLIX class of molecules that were picked up in the TET2 enhancer screen. These compounds are currently in the discovery phase, and we are assessing them for in vitro efficacy as we continue to expand our drug portfolio. All the intellectual property related to these drugs is entirely owned by Hemispherian. Our patent covers the GLIX series of molecules, which is more than 10,000 molecules. We have also filed a patent for a synthesis method for GLIX1. We're currently working to expand our intellectual property and patent portfolio to cover other specific compounds, indications, and synthesis routes. To assess the market opportunity for GLIX series, we are using Avastin for comparison. Avastin is a broad-spectrum anti-cancer drug. Alone GLIX1 has the potential to become a drug with sales similar to Avastin. The complete GLIX series together shows efficacy in a very broad range of indications. Multiple GLIX drugs combined with multiple indications would lead to a significantly larger market opportunity. We are now looking to raise a total of $12.5 million in 2 tranches. This will allow us to complete the phase 1 clinical trial with GLIX1 for the treatment of glioblastoma multiforme. In addition, we will also continue to advance our drugs, including GLIX5, through our development pipeline. The first tranche of $4.7 million will be used to advance GLIX1 through preclinical studies within 18 months. The second tranche of $7.8 million will see GLIX1 advance through a phase 1 clinical trial at the end of year 3. Advancing GLIX1 through a phase 1 clinical trial will give us the opportunity to license GLIX1 or to continue developing GLIX1 through a phase 2 trial. If you're interested and would like to get in contact with us, please call me, book a meeting with me, or send me an email.